Hello everyone, I'm Kumar Sankaran, CEO of Leucine Rich Bio and your host for today. Welcome to the third episode of Microbiome Gazers. The last 15 minutes would be for questions from the audience. Please fill in your questions in the question box. If you're having any trouble with the connectivity, please click on the question mark to get in touch with the Meet support. In today's episode, we would talk about the different technologies used in microbiome profiling and testing, both from a wet lab perspective as well as the bioinformatics. Joining me today is Dr. Debojyoti Dhar, co-founder and director of business development and innovation of Leucine Rich Bio. Deb, I want to ask you a series of questions. To start off with, what is the basic difference between traditional microbiological approaches versus modern technologies with regard to microbiome analysis?
Great. Thanks, Kumar. Great to be back with the new episode of Microbiome Gazers. The topic that we have chosen today for the discussion is very fundamental to microbiome domain. So we'll be very happy to discuss on this aspect in this episode. Yes, Kumar. So, see, the whole story started when Robert Koch in 1885, revolutionized the domain of microbiology by showing that microorganisms can grow. So, the basic principle of traditional microbiology-based, technology is culture. So, you have to culture certain microorganisms to find out more about those microorganisms. And culturing, as you all know, is dependent on a lot of factors. So the factors that play a very important role in deciding whether a particular microorganism will grow in the lab conditions, can be the pH, can be the aerobic or non-aerobic conditions, the nutrients that are required for a particular microorganisms to grow, and so on and so forth. So because of all these variables, it is probably not possible to grow all microorganisms in lab conditions. And when you are dealing with somewhere around 1,000, 2,000, 3,000 species of different microorganisms in your gut, for example, you would not be able to exactly know which microorganisms are there if you only rely on the traditional culture-based technologies. So the new technology that has come up basically in this domain has been in the horizon for, let's say, 20 to 30 years. Initially, the domain looked at the DNA, and there are many various ways of looking at the DNA. Mid 80s, early 90s, mid 90s, the technology that people were using was mostly FISH or fluorescent in situ hybridization, and amplicon based sequencing, because at that point of time, PCR got invented, and hence amplicon based sequencing methodologies came into existence. But things started to change post 2005 when the next generation sequencing technologies, which is a high throughput technology, came into existence. And that's how the shift from the culture based technologies to non-culture based technologies came into existence. And now NGS is the go-to technology, especially when people are looking at microbiome samples from soil to water to even various parts of the human body. So traditionally, that is what the difference is between the traditional microbiological approaches and modern technologies, which rely on mostly the nucleic acids, DNA or RNA. Yes, Kumar.
Thanks, Dave. I mean, that was very insightful. So, what are the non-culture methods available right now?
Yeah. So, as I was discussing earlier, the traditional non-culture based method started with fish, and then it went to the PCR based technologies. Of course, there were some gel-based technologies as well, but those technologies were time-consuming and they were not very insightful. Uh, right now, what major technology that people are utilizing for looking at the microbiome or microbiota rather, basically can be divided into three. One is the again traditional PCR based methods where you use certain primers to amplify certain regions and then sequence and possibly you can sequence either using NGS or using Sangers. The second method, which is also very insightful, is the microarray. There are microarray companies which provide arrays with which can look at different microbial organisms. So microarray is another technology that people use. And the third and the most popular right now is the next generation sequencing technology, which, as we mentioned earlier, is a DNA based high throughput technology, and it is currently ruling the roost in this particular domain. Yes, Kumar.
Let's delve a little more on the next generation sequencing. I think that is gaining a lot of popularity over the world and gained a lot of popularity over the world. In microbiome, especially, I think there are two or three main technologies that are used using next generation sequencing. So I wanted to ask you, what is the difference between 16S rRNA sequencing and what is shotgun whole genome shotgun metagenomics? Now, what are the differences between the two? First of all, what are they, and then what are the differences between the two?
Yeah, when we are talking about next generation sequencing or high throughput sequencing, these are the two major technologies that people will use to identify and classify microorganisms. So let's start with 16S. So 16S rRNA sequencing is something which majority of researchers have been using. Obviously, now the trend is shifting towards whole genome metagenomics sequencing. So, what exactly is 16S rRNA sequencing? So, as we all know, bacteria generally has ribosomes like eukaryotes. The ribosome in bacteria basically have two subunits. The ribosomal subunit is called 70S subunit, and it can be broken down into two subunits, 50S and 30S. In the 30th subunit, there is a operon where you have a 16S rRNA transcript which is present, which has certain conserved regions and certain variable regions. Now, when you so there are basically 9 variable regions, V1 to V9, and you have certain conserved regions. So when you sequence using the conserved regions, primers using the conserved regions, and look at the variable regions, you can actually classify bacteria based on their sequence dissimilarity at those regions. So 16S rRNA sequencing is basically used to look at bacteria, profile bacteria, and create a phylogenetic tree to differentiate between the different bacterial species present in any kind of field. It can be as soil, it can be plant, it can be animal, it can be human. So technically 16S rRNA sequencing looks at only bacteria. Now coming to whole genome shotgun metagenomics sequencing, here you are looking at all the microorganisms, because you are not limited to only a particular region of a particular species, and hence the advantage of whole genome metagenome sequencing is that you are looking at all the microorganisms and you can profile them as per your requirement. Of course, whole genome metagenomic sequencing is, as of now, costlier than 16S rRNA sequencing, but as the trend is shifting towards whole genome metagenomic sequencing, I think the price will dramatically come down. And then the advantages that this particular technology has over 16S rRNA sequencing will be much more felt in times to come. So that's what the basic difference is between these two technologies. But Kumar, when we look at these two technologies, there's another very interesting, let's say, subset of it, which is called metatranscriptome sequencing, which a lot of microbiome, researchers are looking into. So, could you please, share your insights on that particular aspect?
Sure. When we're doing 16S rRNA and when we're doing whole genome metagenomics, in both the starting material is DNA. So a lot of people get confused, especially with the terminology 16S rRNA, that we're doing RNA sequencing at that point. What we are actually doing in 16S rRNA is actually sequencing DNA. Now, when it comes to metatranscriptome, we're looking at RNA sequencing. So we're looking at what is the RNA that is being formed at that point of time, and what is the kind of expression it has. The advantage that it has in research is you're looking at gene expression. You're looking at what kind of genes that are getting expressed with respect to the microorganisms that are there in a particular environment. But as we all know, RNA is unstable. I mean, it does not give you a full picture of what kind of microorganisms are there. It only tells you with respect to the kind of genes that is being transcribed at that point of time. It's a very useful research tool, especially to try and investigate what kind of genes are getting expressed by which organism, but utility beyond that is very limited. So yeah, that's my take on metatranscriptome.
Yes, thanks, Kumar. This is again, as you said rightly, that although it provides a certain snapshot of the microbial, transcriptome, but there are a lot of challenges with respect to this particular, aspect of looking at microbiome using this particular process. Coming to all these processes and next generation sequencing that we have talked about, could you share some insights on the various platforms that are available in the NGS domain? What are the different sequencing platforms that are available?
Sure. So there are, especially with the microbiome, you can use a lot of different sequencing platforms. Most other domains with respect to sequencing is restricted to either one platform or the other. So in microbiome, we can do both the short read sequencing as well as the long read sequencing. What I mean by that is that using next generation sequencing devices, you can look at a shorter DNA fragments, which is called as the short read technology, where we're looking at about 150 base pair DNA sequence. And with the long read technology, you're looking at a much larger DNA fragment, and you're looking at sequencing much larger DNA fragments. In the short read sequencing, I think it is dominated by Illumina, and with all the different Illumina platforms that are available, right from a MiSeq to a HiSeq. Especially when it comes to 16S rRNA sequencing, a lot of researchers swear by the MiSeq platform because it gives you a 300 base pair read length, and based on that, it actually gives you more quality and of data that is coming out. Thermo Fisher also has a good platform for short read sequencing, and the advantages Thermo Fisher has that you have a much less short turnaround time, and especially for clinical samples, I think this is probably the way people are going towards. In the microbiome, I think the biggest advantage right now is in long read technology as well, especially with the likes of PacBio and Oxford Nanopore in the market, providing excellent quality long reads. We're seeing a lot of use cases with respect to the microbiome. With the amount of breakthroughs that are coming through in the long reads, at a given, you can even sequence the entire 16s. Whereas in short reads, you're sequencing portions of the 16s. I mean, sometimes it's restricted to one of the regions in the 16s domain. With a long read, you can sequence the entire 16s, which gives you a much better insight as to what kind of species it is and what potentially can do. In terms of whole genome metagenomics, I think long reads also are giving a lot of advantages, where you're able to identify up to the strain level, which sometimes becomes a little challenging when it comes to short read technologies. So yeah, these are different kind of technologies that are available, and we're seeing more and more platforms coming out, more and more. As they call it, next generation sequencing platforms that are coming out, which might provide a lot more details than what we're getting from the current next generation sequencing platforms.
Yeah. Very insightful opinion on the next generation sequencing platforms. We've spoken about the wet lab part of it quite a bit now. But microbiome is uh, is incomplete without the dry lab, the computational or the analysis part of it, because wet lab will only provide. Yes, Kumar.
Dave, can I? Sorry, Dave. I'm just interrupting you for a second. As we were talking about the sequencing technology. I just wanted to bring up a question that an audience had put up. Is there a benefit of using 16S, 23S genes intergenic region sequencing over 16S rRNA? Is there a practice of doing so?
I'm not too sure about it. I do not know whether that intergenic region sequencing is prevalent or not. What is prevalent is 16S and 16S, the whole 16S with its the conserved region and the variable region part of it. But I'm not too sure about the 23S intergenic region. I'm not too sure about it. Yeah. So coming to the dry lab aspect of it, or what we call as dry lab, is basically the computational part of it, where the analysis is done finally. Because when you the sequencing machine will just provide you a string of A's, T's, G's, and C's. It is the computational part which makes sense of all these strings, right? So could you please share your insights on the various computational tools that are important for microbiome analysis?
Sure. You've got one more letter there, N. We also get a lot of non-identified nucleotides during sequencing. So one of the first steps during sequencing is to identify the nucleotide that is being sequenced. So this is the primary step that needs to be done, which is in most cases done within the machine itself. So either it is, whether it's short read or long read, within the machine itself, the base calling is happening. But there is always scope for improvement in the base calling, and there are many algorithms that are out there which can enhance base calling steps. The primary goal over here is to increase the accuracy of the base calling. I mean, we have to ensure that it is close to 100% accurate when we're doing the base calling. The second part is, so I'm talking about microbiome analysis in terms of looking at the different microorganisms and just identification of microorganisms and their quantity. So in this case, in this regard, the second step would be to bang it against a reference genome data set. When I say bang it, you're comparing it with existing genomic sequences of different microorganisms, and you're seeing what of those microorganisms are actually present in your data, and how much of a particular microorganism is actually present in the data. So this is called as relative abundance. So if microorganism A is present, and 10 of those DNA is present in the sample, and microorganism B, 20 of them is present. So that is the relative abundance it has in the sample. So the secondary step is to identify both in terms of the the name of the species and the strain of the microorganism, as well as the relative number of each of these microorganisms. Post that, based on the sequence, you could also do a little bit of functional analysis. You could look at what kind of genes or proteins the microorganisms could produce. So and do a pathway analysis beyond it to see where in the whole molecular biology ecosystem that this strain of either bacteria or fungus or virus can contribute. Uh, so that is the other final step of analysis that can be done. There is also identification of new species or new strains that can be done through microbiome sequencing, and this boils down to single nucleotide variation between closely related strains to identify the new ones. So these are the kind of analysis that can be done.
Yeah. So, very interesting part of the analysis, as you mentioned, is the functional aspect of it. Uh, of course, functional aspect of it is much more in silico, but still it provides some sort of information on the various pathways that might be important. Uh, but for all these things, you need to have a reference database. So, what are your thoughts on the various reference databases available?
I think that is probably the biggest challenge and the biggest starting point for any of the analysis. Um, so to get the right reference database, to get the right genomic sequences in your reference database, uh, if you go to NCBI and just try to download the reference database dump, you're going to get a lot of noise in it. Because every day researchers are uploading uh, any kind of DNA sequences that they think is a new species, which may or may not be true. So there's a lot of sorting, proofreading, editing that needs to be done before actually accepting something as a as the genome reference for a given species or strain. A lot of times, nomenclature has not yet been decided. I mean, the name of the species has not yet been decided. So if you're doing a microbiome analysis, one of the first things that you will notice is you will get an output file with a lot of undefined species. UNDEF is what you'll see a lot of, because these are species that have been cultured, that have been found, but they have not yet been identified and named as yet. So getting the right databases and actually spending time in going through the database to ensure that the right kind of uh, genome references are put into it is very important. NCBI is a good place to start. 16S, there are a few libraries that are present, which people have done the necessary editing and done the necessary quality check. That's available. But the holy grail is probably NCBI right now to get the right reference base. But this is not a ready to use reference base. You need to spend a little bit of time sorting it and making sure it has the right kind of sequences in it. Um,
Yeah, I have a follow-up question on the database part. You mentioned that NCBI is really big repository of these kind of things. And there are two other for the 16S rRNA sequencing process. We come across green genes and silva. So your thoughts on how these are different from, let's say, the NCBI database down?
what Green Jeans and Silva have done is actually gone through the available 16S rRNA sequences, have sorted them out, have cleaned them out, removed repeated sequences, removed low quality sequences, and only put in identified sequences which is of the highest quality. So if you're new to bioinformatics or if you're new to microbiome analysis, these are the databases to download and do 16S rRNA analysis on, because they have taken that one extra step of ensuring that there is quality of sequences. But it also depends on the use case. If you're trying to identify um, newer organisms and newer kind of bacteria, then you'd probably have to go back and include some of the other 16S sequences that might be left out from these databases. And mind you, these databases are not, although they are updated on a regular basis, they're not updated like on a monthly basis. So there is a lot of challenges in that. But when you're starting off, I think this is probably the best databases to start off with.
As we can all see that the computational part of the microbiome story is very very important, and as we are part of Leucine Rich Bio, one of the pioneers in microbiome domain in the world and especially in South Asia, it was imperative on us to have our own kind of pipelines and other methodologies that we develop. And in that context, Kumar, could you please give your insights on how LRB or Leucine Rich Bio Bio's computational expertise or ability differentiates us from our peers?
Sure, Dave. So, I mean, let's start off with the reference database itself. So, I think uh, we spent close to 6 harrowing months of going through NCBI's sequences and trying to come up with the best policy to identify what kind of reference genomes need to be included in our reference database. And this is not a one-time job. I mean, this needs to get updated every month, because newer sequences are updated or existing sequences are reclassified. So there is undefined sequences suddenly will start getting names. So this is a monthly project that needs to be done. But the first time when we had to download it and get it sorted out, we had to spend a lot of time building this database. And right now, I think we have close to 140,000 microorganisms, and I think very shortly it'll go up to about close to 2 lakh microorganisms. Um, and and from the strain level, I mean, since we do a lot of strain level analysis, from the strain level, what we've observed is um, a reference genome for a particular species. I mean, you can have a strain from an Indian population, and you probably have a strain from a Western population, kind of a thing. So you need to decide what you're choosing and how you're choosing, and for what analysis, what kind of databases that needs to be chosen. So it's not only creating a database repository; it's also building in a computational pipeline that can do all kinds of analysis with respect to reference-based genome identification. Um, then quantity and quality is not the only thing. That's only the first step. I mean, it's only what species has been identified or what strain has been identified. Post that, a lot of analysis needs to be done. So there are two more databases that we have developed. One is um, a microbiome, that is microorganisms that have been identified in particular disease areas. I think we have looked at close to 50,000 clinical and academical research that has been done, papers that have been published worldwide. I think the number has gone beyond 50,000 right now, and from that we have a database which has been manually curated and verified as to what kind of microbiome patterns are seen in what diseases, and which stage of the disease is also very important. I think so. Those kind of things have been identified. Then I think there was a lot of public microbiome data sets that were available, which was used to check whether these kind of patterns were being observed in those public microbiome data sets, just to do a validation of that database. And lastly, but not the least, is probably the uh, is probably the nutrition database, both from human consumption point of view as well as the microbial metabolic pathways point of view. Both, I know we combine it as one database called as NutriBite, but it has both human nutrition as well as microbiome metabolic pathways. Microbiome metabolic pathways, not too much is known. I think about 10% of the microbiome's genes and proteins are being studied, and the chemical reactions are out for that. I mean, a lot of systems biology work has been done on top of that to identify if similar kind of proteins are present, and if bacteria A has one protein whose function has been identified, then in bacteria B, if similar kind of protein is present, the assumption is the function wise it would be the same. So this is one part of that database. The second part is the proper nutrition, and you get to see what is actually present in terms of macronutrients in every vegetable and every fruit and every cereal that we are consuming. So it's easy to identify what is there in, say, potato versus cooked potato in terms of micronutrients, but it's very, very difficult to identify what is actually there in idli, which is like a prepared food. So that is something also that we've taken a little bit of pain to to come up with those steps. The NIN database for Indian food has been very, very helpful in this regard. For Mediterranean and Western food, there's a lot of public information that is available, which needs to be again carefully curated and put into databases. So the nutritional databases, I think, is probably is probably the biggest database right now, and it has a lot of very useful information in terms of what the microbiome does in our gut and how it actually influences when a particular nutrient has been given. So these are some of the databases that are publicly known that Leucine Rich Bio has.
Yeah, so what it means is that microbiome analysis or microbiome domain is a very complicated domain. You can break it down into three aspects, starting from the sample collection, sample nucleic acid extraction from the sample, to the wet lab, to the sequencing part of it, where you have the library preparation and then the sequencing, and post sequencing you have the analysis part of it. And if you look at it, each of these segments has its own complexities and challenges. So the challenge overall of this particular domain is that there are too many variables, and and therefore not anyone and everyone can jump into this, because you need to really understand all the three segments well enough to get into this. And as Kumar has rightly pointed out, the analysis part, the computational part, is the brain behind everything, because anyone and everyone probably can go up to the sequencing part because the machines are available, but what you do after the sequence is very, very important. And unless you have a proper direction to what you are doing with the sequence, and what is the actionable insights that you are trying to provide to the end user, it is not going to be that easy. So Kumar, I think we are done with today's session. If there are any questions, we can take you.
Before I go into one of the questions that was asked, I just want to add to one to your point over there. Though you can't have it as a standalone sequencing and standalone computational analysis. Everything is in conjunction with each other. The kit that is being used for DNA extraction is an important parameter when it comes to the bioinformatics that needs to be performed. So I just wanted to mention this point to the audience, and so on and so forth. What you're doing in library prep, what you're doing, which sequencing are you performing, what sequencing platform are you performing it on. All of that has an impact on the computational analysis that needs to be done. Very simple example. I'll just take DNA extraction as an example. Every kit has a bias towards certain kinds of organisms. Some will get gram-positive strains more, some will get gram-negative strains more. Some will get very low yield in fungus. Where if you try to optimize that yield, then you're losing out on some of the information. So all of this needs to be taken into account into the kind of parameters that needs to be used to do computational analysis. So it is not a very simple plug and play method. It is like you rightly mentioned, there's a lot of variability in all the steps that are performed for microbiome sequencing and analysis, and hence computational analysis needs to be really well thought of. Which is why I don't think it is bioinformatics alone. It is proper computational biology that that needs to be applied for any kind of microbiome analysis. I've just got one question from Dr. Sai. Is the Leucine Rich Bio database available for public? Dave, do you want to give a shot at that?
Yeah, I mean, it's not available for public. This is a database that we have developed, and we are using it for our gut microbiome test that we have launched called Bugspeaks. So right now it is not available for public.
Yeah, but this is especially the reference database. I think we're looking to see how a good SaaS model can be built on top of that, so that other researchers can also leverage on the reference database piece of it. It's in the works, and we'll probably give a shout out once the platform is ready. If I don't see any other questions, so thanks, Dave, for this insightful talk. I'd also want to inform the audience that next Thursday's Microbiome Gazer session is going to be really exciting. We're going to have the world-renowned Dr. Shabnam Kar, who's a functional medicine practitioner based out of Canada, joining Dave in giving a clinician's perspective on the microbiome. We will share the details of the time and the webinar link very soon, and look forward to having all of you all there. Thanks, Dave, for this session.
Thanks, everyone.